Hello. In today's lecture, we will examine the embryonic events that lead to the determination of gender and decide whether it's the human genome or under control of hormones that determine the sexual development of a fetus. First, I think it's important to get some clarity on what constitutes gender. In today's culture, gender has become a somewhat ambiguous term. Classically speaking, gender has most commonly referred to the morphological characteristics that visually denotes one's sex, making someone either male or female. But increasingly, there's evidence to suggest that psychological feelings are contributing to one's gender identity as well. So for today's conversation, we're going to consider gender to include the chromosomal genetics, distinctions of the gonads, morphological traits, and even the psychological aspect of one's gender. From a biological standpoint, gender is assigned at birth. Sexual differentiation occurs at 7 weeks of fetal development. Prior to this point, embryonic tissues are considered bipotential, meaning that the embryonic tissue cannot be morphologically identified as either male or female. If you break down that word, bipotential, you can see that bi is 2 potential possibilities. You have 2 potential possibilities for this embryo to develop into, and that is either male or female. The bipotential internal genitalia consist of 2 pairs of accessory ducts. You have the Wolffian duct, which develops into male accessory organs, and you have the Mullerian ducts, which are female, developing into the fallopian tubes, uterus, and upper part of the vagina. The bipotential tissue is comprised of an outer cortex and an inner medulla. It is these structures that undergo the further differentiation into those classically identifiable male and female structures. The bipotential medulla differentiates into the testes, whereas the bipotential cortex develops into ovarian tissue. Later in this lecture, we will discuss the signal for gonadal differentiation, but for now, that's kind of the understanding we need to know. The basis for sex assignment is programmed by the human genome. During fertilization, 1 haploid egg is joined by 1 haploid sperm. The resulting zygote has a full set of 46 chromosomes with an equal maternal and paternal contribution. So it is an even split of the 2 parents. Only 1 pair of the 46 chromosomes is necessary to assign sex based on a genetic level. The sex chromosomes are the X and Y chromosome. And in the bottom right-hand corner of this screen, you can see a scanning electron micrograph image of both the X and the Y chromosomes, and you can clearly note that one is larger than the other. It is the female or the X chromosome that is significantly larger in size than the male Y chromosome component. Depending on what chromosomes are donated by the parents to the zygote, either a male or female is formed. As you can see at the top, males have 1 X and 1 Y chromosome, whereas females have 2 X chromosomes. Although we won't touch on it specifically in this lecture, it is important to note that only 1 of the 2 X chromosomes of a female will be active. And as we're going to learn in the next slide, it's the SRY gene that is primarily essential to the determination of sex. So the role of genes in male development is a pretty interesting kind of topic to dive into. The SRY gene is primarily recognized with male development as it's Y-linked. In fact, there's a small region on the Y chromosome that has a profound impact on development. The SRY gene directs the gonads to further differentiate. The SRY gene is the signal necessary for the bipotential gonadal medulla tissue to differentiate into the testes. And it does that through a protein called the testes determining factor. It also serves to regulate other male genes, such as the SOX9, the WT1, which is a Wilms tumor protein, and the SF1, a steroidogenic factor that aids in male development. The development of bipotential tissue in the testes is solely influenced by genes. There's no known role of hormones. However, once the testes differentiate, they do secrete hormones that further differentiate and direct male development. So that's a really important distinction to grasp. Hormones don't play a role in getting the testes to form, but once the testes form, they have a significant impact on the development of male parts. The testes secrete 3 hormones. The first is a glycoprotein anti-Mullerian hormone that causes the Mullerian ducts to regress and become not functional. This is the AMH, anti-Mullerian hormone. They also secrete testosterone, which is a well-known steroid hormone necessary for male structures to develop into the epidermis, vas deferens, seminal vesicle, and also aid in the migration of testes from the abdomen to the scrotum. And the last one here is the dihydrotestosterone, DHT. This is a derivative of testosterone, and as you'll see in the image, there's actually an enzyme, the 5-alpha reductase, that is responsible for testosterone becoming DHT derivative. DHT is responsible for the differentiation of external genitalia, and so it makes sense that a lack of the DHT hormone at birth is responsible for hermaphroditism or pseudohermaphroditism, where children appear female at birth, but when they reach puberty, they begin to secrete testosterone, causing a cascade of developmental changes. Kind of as a side note there. Both testosterone and DHT are androgens. They're steroid hormones produced in the gonads under the direction of the adrenal cortex. While male development occurs under the direction of the SRY gene and it's furthered by steroid hormones, female development is a little more fundamental. There is no SRY gene present. Following that, it makes sense that there's no Y chromosome, so females are XX. So if there's no SRY gene present, then the bipotential gonad cortex develops into the ovarian tissues. As you can see by this diagram, if there's no testosterone or resultingly DHT, the external genitalia take on female characteristics and the Wolffian duct degenerates. And without AMH hormone, the Mullerian duct develops into the fallopian tubes, uterus, and upper part of the vagina. So primarily, female development is driven by genes and the lack of sex steroid influence. While there is no direct role of hormones in female embryonic differentiation, it has an important complicated role in female development and reproduction at puberty. Between 8 and 12 years of age, the ovaries will begin secreting estradiol and progesterone. These are both 2 androgens and steroid hormones that are produced. The rise in these circulating hormone levels is responsible for the expression of secondary sex characteristics. And these include the development of breasts, the growth of pubic hair, widening of hips, and the pattern of body fat distribution, particularly around the hips and midsection. So by now you should be seeing that the sexual development of an individual occurs in stages, both embryonic and then again at puberty. At puberty, hormones take on a bigger role in sexual development. As we've discussed, there's no direct hormonal influence on female gonadal development. But in males, however, AMH, testosterone, and DHT influence both gonadal and morphological sexual development. So which is it then that's responsible for the development and assignment of gender? Genes or hormones? As it turns out, the answer is a whole lot more complicated than just picking a side. Both are required at some stage of development. Genomic control does occur first, and it's responsible for our classic definition of male and female, given us how it's the genes and the X and Y chromosomes that kind of initiate this cascade of events later in life. Hormones are, though, critical in the development and responsible for many of the visible changes that kind of identify someone as male or female. We opened this discussion of gender by looking at several factors contributing to gender identity. One of these was a psychological influence, and it's interesting to note that there is evidence to suggest that the exposure of sex hormones during early embryonic development has a facilitative effect on male-typed activities, interests, things they're engaged in, and even spatial ability. And it's been linked to exposure of high levels of prenatal androgens. That has an implication in the behavioral basis of sex that we don't fully understand yet. At the end of the day, it's clear that it's the interplay of genes and hormones that determines gender, and that one cannot work without the other. References.